Hello, today we're going to talk about Anchor 1, Basic Biological Principles. If we look at the first slide here, the slide is talking about the steps of the scientific method. So you should know the steps of the scientific method, 1 being to make an observation, 2 to make an educated guess or a hypothesis, 3 to predict the sequences, and 4 to test the predictions. Now when testing the predictions or performing an experiment, we use a controlled experiment. A controlled experiment consists of 2 groups. You have the control group, is the group that does not receive any special treatment, and you have the experimental group. This is the group that you are performing the experiment with. If you're looking at chocolate affecting test grades, the experimental group would be receiving the chocolate, and the control group would be the group that's not receiving the chocolate. We also need to know the independent variable and the dependent variable when we are doing an experiment. The independent variable is the thing that is different or changed between the 2 groups. It's the factor that you are testing. So if you are testing, does chocolate improve test scores? Chocolate is the independent variable. If you're looking at, does tutoring after school improve the bio-keystone scores? Tutoring would be the independent variable. That's the thing that you're looking at. The dependent variable is the thing that's measured out in the experiment. It depends, literally, on the independent variable. So if we were to say that eating chocolate increases the test scores, the independent variable would be eating chocolate. What depends on chocolate? The test scores. If we say that staying after school for tutoring every day increases the bio-keystone exam scores, the independent variable is staying after school for tutoring. What depends on that? The bio-keystone exam scores. And finally, with the scientific method, we would examine the results or form a conclusion. The cell theory is broken down into 3 parts. Well, if we look at the cell and it's a theory, we can say that all living things are composed of cells. If all living things are composed of cells, where do we get these cells from? They come from other cells through cell reproduction. And finally, the cell theory states that cells are the basic units of structure and function. They are the smallest, all living things have them, and cells can only come from other cells. If we look at the differences here, we have prokaryotic cells on the left, and we have eukaryotic cells on the right. There are some similarities, and there are some differences. If we were to set up a Venn diagram, in the center where there are similarities is that the prokaryotic cell and the eukaryotic cell both have a cell membrane. The prokaryotic cell and the eukaryotic cell both have DNA. And the prokaryotic cell and the eukaryotic cell both have ribosomes. The differences include prokaryotes, no nucleus, whereas eukaryotes have a nucleus. Prokaryotes, no organelles. Eukaryotes have organelles. Prokaryotes are small or single-celled. Eukaryotes are larger or multicellular. Prokaryotes, example is bacteria. Eukaryotes, the example is humans or anything other than the bacteria. Organelle is that tiny little organ that does specific functions in the cell. If we look at this next slide, we're going to go through the cell and identify the different organelles. The first organelle is the cell membrane. And the cell membrane is also given the name plasma membrane. And the plasma membrane is the outermost area. If we look here, the plasma membrane, the cell membrane goes around the cell. And its function is to let certain things in, like oxygen, gases, food, and it lets certain things out, the waste products, CO2, water, etc. Then next, we have the cytoplasm. It's the region inside the cell, contains all the organelles, and it's a jelly-like substance. So it's really everything inside the cell except for the nucleus. The next slide is we talk about the nucleus. The nucleus stores genetic information, and it also controls the cell's activities. So it has really two jobs: to store the DNA, and it controls all the activities of the cell. The ribosomes make proteins, and they can be found freely or they can be stuck on the endoplasmic reticulum. If we look at the endoplasmic reticulum, there are two kinds. There is the rough endoplasmic reticulum, and it is rough because it has ribosomes. And if we know from above, ribosomes are involved in making protein. So they have the little bumps. The smooth endoplasmic reticulum, they don't have the bumps or the ribosome, and its job is to make steroids and lipids, as well as breaking down toxic substances. The next slide is the Golgi apparatus. The Golgi apparatus is a system of membranes and stacks like a post office. What does a post office do? It packages. Two things that it packages are from the ER. If we can call the rough ER is involved with proteins, and the smooth ER is involved in lipids. So therefore, the Golgi apparatus packages lipids and proteins. The mitochondrion, or mitochondria, is basically known for making ATP. The next slide is lysosomes. Lysosomes has digestive enzymes that break it down. You can think of like lysosomes like Lysol. It basically kills the bacteria and viruses, etc. Lysosomes, digestive enzymes. Cytoskeleton, we see the word skeleton involved here. So like our skeleton, it basically supports the shape of the cell. We have cilia and flagella, and both of these involve movement. The cilia are short and large numbers, and the flagella is one long tail like the sperm. So if we look at the next slide, we can see the cilia here, these tiny little hairs, and then we see the flagella has the long tails. And if we look at the organelles, here's the mitochondria, which makes ATP. You have here the smooth ER, which is involved with lipids. The rough ER, which has the dots, the ribosomes, it's involved in proteins. You have the Golgi apparatus, which is involved in packaging the proteins from the rough ER and the lipids from the smooth ER. They both go here before it's sent out of the cell. The free ribosomes are these dots little here, and they are involved in protein synthesis. The nucleus, this center here, which stores the nucleus and controls the cell. The lysosome is involved in breaking down the digestive enzymes. And we have the cytoskeleton for shape. And we have the plasma membrane here, which controls what goes into and out of the cell. The next slide is involving plants. Plants are different. They have all the same organelles as an eukaryotic cell, than an animal cell, except that it has three special things. One being a cell wall. If you notice what is a wall, what's the purpose of the wall? It's to help support the plant. It's found on the outermost, so the wall is the outside, and then you would have the membrane, the cell membrane, on the inner part. So the membrane is inside of the cell wall. The next thing that a plant has is a central vacuole. If you look here, the central vacuole, it's the center, and it basically stores water and waste. And finally, the other C word is chloroplast. You can see here's the chloroplast, and we know that the function of the chloroplast is basically to do photosynthesis. So the 3 C's: cell wall, central vacuole, chloroplast. The final slide is that we are going to talk about from unicellular to multicellular. What is the flow? Organelles are organized into cells. Cells form tissues. Tissues form organs. Organs form organ systems. And your organ system forms an organism. If we go backwards, we could say you are made up of organ systems. The organ system, like the digestive system, has organs in it, like your stomach or your intestine. The stomach is made up of tissues, and your stomach tissue can be broken down into cells. That concludes Anchor 1 information in a nutshell. It's not the total notes, but you get an idea of the most important parts of Anchor 1.